morning, good afternoon, good evening, and welcome to 1% Better, where we really take a look at hidden stories in sport. I'm your host for this week, Dr. David Lippman. Joining me today is former guest, current guest host, following her beautiful piano presentation that she sent us, or beautiful piano playing she sent us to put on social, soon to be Dr. Kristi Storoschuk. Thank you for joining us, Kristi.
Oh, it is such a pleasure to be here. I'm really happy I was the chosen applicant due to my performance on the piano. I think I was the only applicant, but I'm still very blessed.
Only applicant, but not the only option. How's that?
Oh, okay. Thank you.
So we'll go that way. So we're going to flip this a little bit. The medical question is going to you. What is the only organ in the body that doesn't have any mitochondria?
Organ?
It's not really an organ, to be fair.
Red blood cells?
Yeah, that's the
Okay.
answer. Yeah, It is the red blood cell. I told you you'd get it. So yeah, red blood cells don't have any mitochondria, which is cool. Fun fact. And they only last about 120 days. Short life cycle. So we are supported on this podcast by Precision Fuel and Hydration. So I wanted to say thank you to Precision for that. We do have a question upcoming from our CMO, Spencer Lauderbeck. We threw forward to this question last week about his use of chews and their other nutrition products. So if you want to be like Spencer, you want to get some Precision Fuel and Hydration, and you want a discount, you can use our code OPB2025 or follow the links in our show notes, which do go to their website with the code already applied. That is on your first purchase only. Kristi, you've just come back from a trip to Madeira, one of my favorite islands in the world, one of my favorite places in the world. How was that?
Well, it's now my favorite place in the world. That was the best trip I've ever been on. And also, I just want to put it out there. If you see an epic trip on Instagram, book it, because that was my experience. I saw the through hike from west end to east end of the island of Madeira on Instagram, booked it immediately, and my boyfriend and I packed our tent and we headed out. We booked campsites well in advance, actually, and camped along the island, crossing by foot, and it was unbelievable. It was so amazing.
That is how I ended up there. My first trip to Europe, I'd seen this Instagram account that followed me, Go Trail Madeira. Shout out to Daniel there. And then I was like, I'm going to go do that. I'm going to do some running there. And then I was telling you offline that I met my now wife, or she came over to do those trips with me before she was my even girlfriend at the time. So that was really cool. So fond memories of Madeira. And as another update, where are you at with your PhD? I don't know if we asked you this on the previous podcast when we had you on as a guest, but I think it's helpful there. Where are you at?
Okay, quick update. I need not a few. I guess it's a lot. 25 more participants to finish data collection, which seems like a lot, but it's actually a little when I have over 100 total. And then just head down in the wet lab analysis, and then head down again, writing up my dissertation. And I hope to graduate within the next, 6 months.
Okay. And so you're taking muscle biopsy samples. Is that correct?
Yes. So okay. I need to convince 25 more people to get a muscle biopsy, and then I'll have all my samples ready to analyze.
That's awesome. I'm excited about that. And what about training? Where are you at with training? What's happening? I mean, maybe you don't train, you exercise historically, you've told me. So where are you at between them?
Right. I don't train, I exercise frequently. But I have High Rocks coming up. I signed up for the Toronto High Rocks, and I did the pro women division, which I may be in over myself with because the weights are pretty brutal.
I was going to say, there's not a lot of you, and part of shifting a sled, it helps having body weight behind you. So I'm interested to see how you go here.
I know. I really have no idea what to expect. I just want to finish, but I didn't want to sign up for the scaled version of it. Like, if I'm going to do it, I want to do it properly, and we'll just see what happens. I don't know.
So are you going to train for it, or are you just going to keep doing what you're doing?
Just keep doing what I'm doing.
Okay. Well, maybe do some sled work is my takeaway here. Good news, you're a very good runner, and it's a runner's sport. The bad news is, I've heard, I'm not sure it's the truth, but I've heard doing high rocks gives you a shirt allergy, and you're not really allowed to wear shirts around high rocks. So you may develop a shirt allergy, you may not.
Well, surely I will be covered.
Hopefully so. But we're also supported on this podcast by Stride. And so have you come across Stride power meters? The things that go on the shoe, give you running power? No? Mm-mm. Okay. So these give you, running power. So they go on your shoe. I wear them. I wear them on both shoes. So they can give you kinematic data for the differences between right and left leg, which is really interesting, and different strides and how they deteriorate across a run. So super helpful in the injury context. And then also gives you an idea of metabolic power for running. So you can use these to do things like select which super shoe is best for you because your power goes down for the same pace, right? So the metabolic power is reduced to create the same output.
That's
So
so interesting. So yeah, like shoe choice, it would
correct.
help you.
Yeah, so shoe choice helps a lot. It helps with some pacing when there's hills or wind or things like that because it's a relative constant. So, and it gives you a nice metric to triangulate with. So you can kind of use like heart rate and pace and then power and then perception as well. So sort of add it into your little stack of what you use there. So
That's super interesting.
yeah, as you become a marathoner, you're going to buy a pair, I can see. So let me know. I can put you in touch with the Stride folks. But we do appreciate Stride for supporting the podcast. They did join a little while ago, and we've been enjoying exploring Stride and its metrics and sharing them with people like yourself. So thank you for being part of that and
I'm
part of the
interested.
community. Yeah. It's good for data. I'll tell you that much. I can show you some stuff offline. But wanted to get to some community shout outs. We had a listener question from our CMO, our Chief Meme Officer, Spencer Lauderback, that we mentioned before. So his question is, I recently did a 37-mile run at zone 1 and zone 2 effort, well below LT1. Should I have carb loaded before that? Question mark. I did not, as I was assuming the majority of my energy was going to come from fat. I took 30 grams per hour of carbs from Precision Fuel and Hydration Chews, thinking I wouldn't need any more carbs than that for such a low intensity effort. What are your thoughts on that? What would you do if you were doing something similar? So I'm going to throw it to you first. Chris, you have strong opinion. I have strong opinions here. Now you've got good experience here. So I'm interested in what your thoughts were, and then we can sort of discuss some things and throw some things around.
I mean, I definitely don't have strong opinions. But I would say like you did the run. So how did it feel? I think that's the most important thing to take into context.
Yep.
I don't have any answers. I'm really not that familiar with the grams per hour carbohydrates and then taking in intensity. But I will say that we have enough zone 2 literature to provide evidence that glycogen depletion is significant with zone 2. Once you hit the 2-hour mark and beyond, you're significantly glycogen depleted. And so carbs are obviously going to play a role. And 37 miles is a huge run. So I'm assuming it's going beyond 2 hours. In that case, then you would want to figure out your carbohydrate strategy. But if 30 grams per hour worked and you got through it and you felt good and were able to recover, I feel like that's the most important piece of information to take into context. And then beyond that, I don't have anything specific, but I'll let you take it from there.
Yeah, it was about, from memory, it's like 6 and a bit hours, maybe 6.5 hours. So it was quite a long time. Maybe I'm wrong there. Apologies if I've misquoted that. I mean, so do you need a carb load? My answer here is like, it depends on whether you're trying to optimize your performance or not. If you're just trying to get the run done, probably don't need a carbohydrate load. If this is a race and you really want to see the best you can do, carbohydrate loading, would suggest that that's helpful in that context. To your point, though, increasing our carbohydrate stores does mean we burn more carbs. So it's just kind of a bit of it's still a net benefit, but you will burn more carbs doing that. Similarly, you'll burn more carbs by taking in carbs. But the question is, how much do you need per hour? Yeah, it's a good question. Look, there's some literature. We had Andrew Kutnick, who I know you know, on the podcast, talking about a paper they released looking at 10 grams an hour to try and the working theory is basically you need to maintain blood glucose, and the liver provides some glycogen for that, and then you can top that up and be done. Because to your point, muscle glycogen, which is what you were alluding to, is depleted at an intensity dependent rate, regardless of anything else you do. So the best you can do is hope to maintain liver and blood glucose, liver glycogen and blood glucose, and you're probably not going to maintain liver unless you're taking in many grams an hour. So I think your point was a very good one, though, which is how'd you feel? How'd you go? If it was all right, then maybe you did enough. Is it optimal? Maybe not. Is it possible? Certainly, it was done, right? So then the question is, how did you recover off it? Which is another one you posed, which I think is good. And that's, to be honest, a lot of the carbohydrate research and this 120 grams an hour, if we look at where that came from a paper by Aitor Viribay looking at a mountain marathon. And all they showed was that markers of recovery and muscle damage the next day were better. They didn't show an increased performance. They showed that there was some signal that things were better the next day. So it's worthwhile understanding that, firstly, that's where the 120 grams an hour came from. But secondly, and more importantly, sometimes carbohydrate intake is about recovery, not about performance at the time. So I think if you recover well off this, then you're fine. I mean, listen, I've done, and I know you've probably done some, knowing you, Kristi, like a bunch of running fasted, right? With zero intake or very little intake. And it's certainly possible. And, again, you're not going to perform optimally, but yeah, you're okay doing it. What's the longest run you've done fasted, Kristi?
Ooh, I honestly probably just a half marathon.
Just a half marathon. That's a long way. That's a long way. That's what, 90 minutes thereabouts.
Um, just over, unfortunately.
Yeah, yeah. So 90 minutes. I used to manage about up to like 3 hours fasted. But I would feel pretty rough towards the end of it and for the rest of the day. So I would not recommend that. But the context here is that you certainly need to think about context of where you are and what the goals are around that run in the season, in your program, in the month, week, day. And then thinking about other things. So yeah, carb loading, something more than 8 grams per kilo of body weight, more like 10 to 12 for 24 to 36 hours. So that's a lot of carbs, and you eat hard to do that. That is a lot. But you could certainly do something more contemporary, what they call carb periodization, which is when you're going to use more carbs, you bump up your carb intake a little bit. So, and then lower it on days where you sort of don't need them as much, right? So that's certainly possibility. In terms of the 30 grams an hour, probably okay. Could probably go a little bit higher. I mean, the research suggests up to 90 for these sort of durations. But again, intensity is a variable, as probably is body weight, although that's not really well supported. But you certainly would burn more as a heavier person running the same pace, right? There's more energy required to do that. So it's worthwhile consider it.
Would up to the 90 grams per hour, like, would it depend on intensity though? Like, if you're just going zone 1, zone 2,
Yeah,
then the grams per hour would be less.
I agree. The guidelines don't really suggest that, but I think practitioners would agree with you, and I tend to agree with you, which is like you can't tell me that me running for 3 hours zone 1 is the same as me running for 3 hours at zone 3. Like it just
Yeah,
isn't the
exactly.
same. So your carb burn's different, and the requirements are different. So yeah, I tend to agree with you. And I don't think Spencer's done himself a mischief doing this at 30 grams an hour. So particularly if you're more experienced, one thing I'd say is like experience comes into this, because part of experience is A, knowing your body and when it's going pear shaped, and B, fat ox rates, right? So he was saying he thinks he can support it burning mostly fat. It's true to a degree, but your ability to burn fat is somewhat contingent on habitual diet and then training history and all those other things, right? So yeah, it not always. And the other thing that I think I've sort of beaten the drum on enough, and hopefully it's getting into people's head, is the difference between relative and absolute intensities. Just because your relative intensity is low doesn't mean that your absolute intensity is low, and therefore you may still be burning a lot of carbs even though it's a small percentage. So 5% of a big number is a big number.
Right. If your zone 2 is running
Exactly.
like 430 kilometers, then
Exactly. So you might be burning a lot of carbs, so you may need more there. But again, you don't have to put everything in your mouth that you're burning, right? So if you're burning, let's say you're burning 100 grams an hour, you don't have to put 100 grams an hour in your mouth, right? So that's also worth remembering when I'm talking about carbs. I think it's done a pretty good job there. So hopefully that answers your question, Spencer. I appreciate your input there, Kristi, as well. So let's look at some stuff that's come across my desk. So we've talked a lot about heat on this podcast, heat training, etc. It was one of our most popular episodes, and then Kristi joined us as a guest, and then broke some of those records. And then we had Clara Kopperberg join us, and her episode took off. So sorry to tell you, Kristi, you're not
oh
quite top
man.
anymore. But you were like edging towards the top. You're now at number 2.
That's okay.
But I thought this paper would be good to discuss. It really looks at some of the changes that happen with heat and implications for, I think, training and racing. Because, we are currently, you and I are both in the Northern Hemisphere, we're heading into the colder months, but the Southern Hemisphere, where many of our listeners are heading into the warmer months. So the paper was called The Effect of Heat Stress and Dehydration on Carbohydrate Use During Endurance Exercise, a Systematic Review and Meta-Analysis. Give us 1 minute on what a systematic review and meta-analysis is, Kristi. I've sort of mentioned this before, but you are in the PhD world. You've got a much better understanding of stuff than I do. So give us an
Yeah.
idea.
So to conduct a systematic review, it's a very rigorous design. You follow a very strict guidelines of what you have to do for it to be considered a systematic review. So you have very specific search terms that are consistent among the databases that you're searching. So you basically have a preset guideline before you even conduct the review. You have your search terms designed and everything, and you put those search terms into a database. So something like PubMed, for example, and you take all of the results from that search and you basically screen them all. So you look at every single study that was captured with that search and you pick the ones that fit your inclusion criteria, which should be pre-registered beforehand. And then from there, you're just basically filtering through these certain steps until you get the studies that you're looking for. So a systematic review is a really highly rigorous and well-regarded type of study because it does go through these guidelines and should capture, if your search terms are done correctly, every single study that has looked at the question that you're asking for. So and then when that's the systematic review part of it. And then the meta-analysis is taking all the results from every single study captured that fits your inclusion criteria and then running the stats on it that's the meta-analysis.
And they often try and group those results together if they can in the meta-analysis. Is that true? To try and create, say, going from 5 samples of 30 to a collective sample of 150. Is that fair to say?
Right. Yeah. So for it to be considered, to even run a meta-analysis, they have to be a bit homogenous. They can't be the study design can't be so heterogeneous that you can't run any stats because they're just so different from each other. And that happens sometimes. So you'll see just a systematic review and you won't see that and meta-analysis in the title.
Yeah. Awesome. Super helpful. So this systematic review looked at heat stress, so hot compared to temperate conditions. And again, these are the things that they would have to have put into their systematic review, and dehydration. So dehydrated compared to hydrated status. So they looked at, is it hot or temperate, and are people hydrated or dehydrated? And they looked at respiratory exchange ratio, which is a way, so we measure inspired and expired gases, right? You put the mask on, look like Bane. We're measuring oxygen in and carbon dioxide out. And this is what we call your respiratory exchange ratio. And this gives us an idea, not an exact measurement, which is key, of what you're burning, carbs and fat, etc. They also looked at carb oxidation and they looked at glycogen use. And glycogen use is, I don't know how they actually looked at that. I assume they used measures. They did something to measure glycogen because otherwise you're just guessing. So you're having a look at that. So they had 51 studies that they found, 502 participants between those, 31 females, which is 6%. So this is particularly concerning given that women respond differently to heat and heat loads. So this is I'm a little bit concerned about some of these results in terms of application to women. But what they did find was that carbohydrate oxidation and glycogen use were higher in hotter conditions. And they were also higher in a dehydrated state, but that may only be in hot conditions because things were pretty inconsistent there.
Yeah, so that's interesting. So dehydration only matters when it's hot?
Probably. It probably is more of a factor. So one thing to remember when it's hot is that we are peripherally dilating, right? So that we are shunting blood towards the skin to cool off, right? We want to use that to cool off. And if it's cooler, you're not doing that. You're going to shunt blood centrally to keep yourself warm. And so the problem of dehydration, if we call it a certain percentage of your blood volume or body weight, if you have less of that and we're shunting it peripherally, we're increasing the space in which it can exist in terms of open capillary beds. So your blood pressure drops more and more. So your performance is affected more and more and more, so to speak, because it's a compounding problem.
Interesting.
So I think, this is interesting for me. One of the things I mentioned here or thought about here was that it's a bit of a wicked problem because in the heat, you burn more carbs, but you also have a higher risk of GI upset as a result of the heat. And then if you're trying to take in more carbs, you have an increased risk of GI upset. So you end up with this real significant impairment in performance and a higher risk of, I call it catastrophic failures. Maybe it's a bit harsher word, catastrophic, but if you end up having to run to the toilet, that may well be the end of your race, right? And maybe you have to stop running or triathloning or whatever you're doing. So what I'm getting at here is that you may be trying to bump things up, but you may end up in a bit of a wicked problem. So what I would suggest, particularly if you're training for summer stuff, is gut training. Really, really important. You need to understand what you can take in stressful conditions. So I would be, I did a bit of this before one of my marathons, I was doing about 110 grams an hour so that I could be comfortable at 90 in the race, because the stress doesn't help either in the race.
What if you became more heat adapted, like expose yourself to heat stress?
It's certainly helpful. I think that's a no-brainer. You just have to do it. I think you have to do, and listen, so there's a difference between heat adaptation, which is something that we try and actively do, and heat acclimation, which is something that happens naturally. So if you go through a summer, you'll acclimate to heat, whereas heat adaptation and heat training is sort of artificial stimulus. So I would be doing both if I was racing in summer or anything like that as best I could. Now, in Europe, you can't really heat acclimate because it doesn't get that bad unless you're in the south of Europe. So you're sort of having to heat adapt. But I would certainly be doing that, and I'd be trying my best to do some of those as active sessions. So you've got active versus passive, which is passive sit in the sauna, active run in the sauna, or run wearing extra clothing. I would be trying to do that with high rates of fueling as well, because A, you're going to be burning it, right, as we sort of alluded to here, but more importantly, you're going to train your gut because, again, it's stressful.
Okay, so I've seen you running in a suit. Is that the heat? Is that for heat stress?
Hazmat suit, yeah. So there's a number of ways you can do this. Companies sell them, all sorts of things. What I do with that suit is you can just wear, and there's papers on this, but on what they wear, but it's like multiple layers, including like rain jackets and stuff. And it just gets a bit much and you feel like you're running like the Michelin Man. So I ended up buying, it's just a paint suit, which is like pretty much, it doesn't breathe. And so I wear tights and then a warm long sleeve shirt underneath that with like a beanie and gloves. And then I put that on as well. And that creates a real horrible microclimate. So
that's hilarious.
I was doing it running along the river here and I got some serious looks like, what's this guy running from? Because like, it looks like a hazmat suit. They're like, should I be running? What's this guy doing? So
it's so funny.
Yeah, it's it's not for the passion joggers around us. So let's put it that way. But it works and it's really unpleasant. I'll be honest. And it's like really, it cooks you. You need to be really careful, drink enough water, all those things. So yeah, you had some questions around some of the mechanisms here. Is that right?
Yeah, I was just trying to understand why we would burn more carbs if we're hot, and so we discussed this off air. But the blood flow being one of the primary hypotheses of what's happening. So I guess when we're hot, blood flow is trying to go to the skin, the surface of the skin, cool us down. And that means that we're getting less blood flow and oxygen delivery to the working muscle, which will increase glycolytic flux because now our aerobic metabolism should be a bit limited. And then therefore we're activating carbohydrate metabolism, burning more muscle glycogen.
And then probably on top of that, like sympathetic nervous system tone is probably higher, which will make us burn more carbs and more adrenaline, and then probably like more cortisol as well, both of which will upregulate carbohydrate burning and glycogen use. Right. So I'd say those are probably some of the mechanisms, but I'm sure there are smarter people who are tearing their headphones out and throwing them at the ground at the moment. Listening to
these are the mechanisms these were discussed in the paper. They had three basically primary hypotheses.
Yeah. So if you're an expert, feel free to DM us on Instagram. Let us know. You can come on and talk about it. We'd love to hear from you.
But I guess the takeaway then, practical takeaway from this being that you're going to need more carbohydrates. You're burning more if you're hot, and thereby potentially dehydrated too.
So I think it's harder refueling in summer. So like more carb intake post training in summer, I think is probably, or hot conditions, heat training as well. I think if you're going to race, you need to be prepped for that. And so it's probably some gut training to be prepared for increased stress. And again, race day is going to be more stressful. So you can have less blood flow to the gut. So you need to be prepping almost excess of what you need to perform well on race day. And then be wary that you may not perform as well in the heat, right? Do your heat training, do your heat prep, but you just may have to drop down the carbs and you may have to drop down performance. That's something that just happens. I'm sorry. But
hopefully it's happening to everyone, so it's all relative.
Yeah, and if you prep for it, you'll be better off prepped, right? So you'll perform relatively better in the field. That's 100% it. Like they say, the more you sweat in training, the less you bleed in battle. So it's worthwhile thinking about that. Yeah, it's one of the things I used to say to try and get people to come to training. I don't know if it worked. But something you're interested in, I know you're interested in maternal health. I think your sister does a bit with maternal health as well, from memory. So I found an article that I thought was super interesting for anybody who's an athlete, but certainly for those who are thinking about having children and who are athletes themselves. So this was called Effects of Sports Disciplines on Offspring Sex Ratio in Elite Athletes, an observational study. So again, observational study, they didn't intervene here, right? They just looked at what's happening. They did not tweak anything. They just looked at rates here. So I guess starting off with some definitions, sex ratio is the ratio of male to female births. It's a demographic indicator, and it's close to 1.04 to 1.05 in the worldwide population. And that varies depending on environmental conditions. So basically there are more males born than females in the sort of worldwide population at a rate of, 4 to 5 per 100, right?
Yeah.
And then there's this thing of like elite sport exposes athletes to several factors known to impact sex ratio. And I didn't know these to be the case that well. In particular, high physiological training loads, psychological loads, so that's like life stresses, and then other levels of stress. So that's interesting. I didn't realize that would be as big a factor here. But they looked at 2,132 athletes from 45 sports disciplines. And this included 399, which was 18%, were females, and then 1,597, which was 74.9%. Athletes were selected in their senior national team or international competitions, right? So like high level athletes here. Overall, they analyzed the data from what was almost 3,000 children, 2,995, with a mean sex ratio of 0.981. So it's 6 more girls born per 100 births. So it's almost the inverse of what you'd see in society, in free living society or world's population, where it's 4 to 5 more males. You're talking about 6 more females. So it's a big swing there. And they had observed within the sports values ranging from 0.548 sex ratio in middle and long distance running, which is significant, right, to 1.275 in tennis. So in tennis players, many more male children. In middle to long distance is many more female children. Now this is where it got really interesting. Female athletes had a significantly reduced sex ratio compared to their male counterparts, 0.845 to 1.016 respectively. So that's saying female athletes had more female babies than their male counterparts. And there was no difference in sex ratio of children born during versus after the active period. So this has got nothing to do with their performance, like their training specifically at the time, whether they're retired or not. And then endurance athletes are more likely to have daughters, as we sort of alluded to before. And then the probability of, they call it siring. I don't love this. Siring's what animals do, but the probability of siring daughters was strongly enhanced in female athletes who gave birth during their professional sporting career, apparently. So stress seems to lead to more female births. What do you think of this? I thought you'd find this fascinating.
Yeah, it was super interesting. It was interesting just to read what the authors had to say about just the lifestyle of a female athlete versus a male athlete. So the demands, the psychological stress of a female athlete being above that of a male athlete. So also just having to take time off sport to be pregnant and give birth that a male doesn't have to do, the different roles that a female takes on when they have a child that would induce stress and introduce stress that a male might not have to deal with during their professional career. And that stress, that psychological, physiological stress, that impacts the sex ratio, which that was very interesting to me. I didn't really realize that, but and then that this is manifesting in the data. So with females being more likely to have a female daughter or female offspring than the male athletes. So it's interesting. And also maybe this implies that females are just under more stress in general than male athletes.
Yeah, I mean, it would make sense, right? Like just the concerns around pregnancy is enough. Am I going to get pregnant? How do I mitigate that? If I am getting pregnant, what does that mean for my career? When should I get pregnant? Right? All those questions are real. And it's 2025 and that might sound foreign, but if you think back 10 years, there were stories of people losing contracts with it. You know, Alison Felix was a high profile case with Nike. She lost it like she was going to give birth and Nike sort of apparently cut her for that reason. So, you know, pregnancy clauses in contracts are new. They're not old, they're new. So it's worthwhile considering that's a real stressful aspect. And then there's all the other factors. Now, I need to get somebody on to talk about this, but there are enough women performing really highly, particularly in endurance sports post-pregnancy, that I wish to talk to somebody about this. So if there's anybody you know who knows this and you're in our ecosystem, please reach out to us. I'm very interested to talk about this. There's lots of theories around it as to why that may be the case. Psychological, physiological, all those things. But I find it to be a fascinating observation. And I've spoken to many of these women and they tend to agree with that being a true finding. But maybe that's wishful thinking on their part. I don't know. So
performance enhancing childbirth.
Yeah. Yeah. I mean, from a physiological standpoint, talking about blood volumes and pain tolerance and all those things. And then psychologically, you can talk about like perspective and all that stuff. I don't know. But I'm interested in it. I find this to be interesting. I sent this to a friend who's just found out he's going to have a little girl. So I'm excited for him to have a child and it be a little girl. But I'm excited that this is the case. So we talked about it a bit off air. The reason this may be the case is to do with basically like survival of a species. During a stressful period, you need more females because they can only like mother one child at a time, maybe twins or triplets, right? Whereas males can father many children in quick succession. So in terms of like how this would have evolved, that would be one of the evolutionary pressures that might have created this during periods of stress.
Yeah. One thing that I would point out too is that they only included like 18% of the data included in this was from females, and the other
Yeah.
80% was from male athletes. And I wonder if it is just that female athletes, one, might be infertile because they're athletes, and that's obviously a big risk is
Yep.
the hormonal imbalances. And yeah. And so it's just interesting that this population was mostly males.
Yeah, and also it's going to give you stronger data or stronger looking data in women. It's going to slant the data to see things that may not be real, right? So it's worthwhile noting that. I mean, I do think there's maybe the ratios are out for the proportions of like females having female children, but I do think that the probably what we're seeing is directionally correct. It's just what's the significance of that direction. So I do
Yeah.
think the stress will increase female births. It's just how much is probably worth questioning. And are the numbers big enough, right? Do we have hundreds of thousands of births? No. Okay, let's like take it with a grain of salt. There is some data on, I believe, on anesthetists and the gases and the passive gases they get driving more female births as well, or female children. So
Oh my gosh.
yeah, and it's very common for anesthetists to have female daughters. So, daughters, female children. So there is some data on that. And I have a friend who's an anesthetist who has a daughter. So he happens to run a lot as well. So there's that. But now we're on to something that's in your wheelhouse, a bit more in your wheelhouse. Sometimes extremes inform the norms, so to speak, I reckon. And specifically in this case, an extreme example, more relatively extreme, fasting for 7 days, gives us some insight into what we should expect from maybe low carb intake or energy deficits to a degree. So this paper was called Effects of 7 Days Fasting on Physical Performance, Metabolic Adaptation During Exercise in Humans. So they took 13 participants, 7 males, 6 females, for 7 days. They started with 15, actually, but 2 dropped out. They looked at maximum isometric and isokinetic strength. They looked at VO2 max. They looked at, they used CGMs, continuous glucose monitors, and looked at glucose levels. And they looked at DEXA scans, which are basically big old x-rays of the whole body to look at body fat composition, bone mineral density, etc. And then they also use some really cool stuff, which I was really interested in. They use nitrogen balance, which is like an advanced method of understanding how much protein you're turning over. Yeah, that's what they looked at. And they looked at some exercise measures as well, and they sort of went from there. So we can sort of talk about some of their findings and go from there. So in this, let's talk some of their findings. So their isometric strength, or their strength remained unchanged, while their VO2 max went down 13%. The average body weight went down. Yep. Please.
But their relative VO2 max went down less because they lost body weight.
Yeah. So that's a key point here. So let's talk about that. So VO2 max is usually measured, or most people understand VO2 max as a number that's like 50, 60, 70. That is relative VO2 max. That is milliliters per minute per kilogram of body weight. So if that kilogram number changes, your relative VO2 max can change without your absolute VO2 max changing. So your absolute is liters per minute, right? So they don't normalize for body weight. And that's because body size impacts your lung size and therefore the amount of oxygen you can get in, right? So rowers have historically done it without body weight because your body weight's not really that relevant a factor in rowing because you don't have to support it. It's in a boat, and they have huge ones. They have like 7 liters because they're such tall humans. Whereas in URI, it's not like that. So your point's well made here, which is their VO2 max went down, but because body weight went down as well, relative VO2 max doesn't change as much. And the relative does change, but the absolute doesn't change as much. No.
Yeah. I mean, it still goes down, but it went down like 7% instead of 13% or something in that ballpark.
Yeah. That's a good point. Average body weight decreased 5.8 kilograms, plus or minus 3 kilos. That's like, let's call that 14 pounds, thereabouts.
That's a lot.
That's a ton. That goes down a lot. And some of that's going to be glycogen, and some of that's going to be water that goes with the glycogen, surely, but not that much. We're talking maybe that's a kilo if we're lucky. So it's still 80% of that's probably something else. So the DEXA we talked about, so they looked at things. And so after 7 days of fasting, lean mass dropped by 4.6 kilos, which is an 8% decrease. Now, lean mass isn't all muscle. We tend to think it is, but it'll be all sorts of stuff. It'll be maybe a bit of bone, maybe a bit of ligament, all that sort of stuff, bit of organ, maybe like the liver looks smaller because the glycogen content is less. So all that stuff will factor into it. They looked at the extremities, and this is generally where you look at lean muscle mass because you can only really get that from the extremities. So that looked like 1.7 kilo plus or minus half a kilo reduction in lean mass. So that's 6% reduction in arms and legs. That's quite significant. Fat mass was reduced by 5.7%. In the extremities, fat mass in the body was 1.4 kilos. So that's 8%. And then a reduction in fat mass was associated with the body weight. So that would make sense, and lean mass prior to fasting. So the more fat you had to lose, the more you lost. Bone mass remained unchanged, which is pretty important and would make sense in a short term if you're loading your bones and all those things. If we put them in bed for a week, it would go down even if they weren't fasting.
Yeah, so that was a difference between this and like prior studies that have looked at fasting and bed rest, is that things declined way more rapidly if you're just laying in bed versus these individuals were free living and able to move around, and I guess they were able to engage in physical activity, which probably was a significant mitigator of both muscle loss and obviously the bone loss and the strength loss, like the lack of strength loss that they observed in these participants.
Yeah, for sure. I think it would be cool to see like a 4-way design where you've had fasted and unfasted groups, bed rest versus exercise, and see what happened there, where you could start to try and tease out, is this a result of inactivity or fasting, right? And we can start to tease it out where you've got a 4x4 where some are fasting and doing activity, some are bed rest, and then some are not fasting and some are and doing activity and some are bed rest. So, and you'd start to see there the impacts, because I think to your point, a lot of what we think we know about stuff is to do with rest more than it is to do with fasting.
But 7 days is a pretty extreme fast. So
yeah,
and
I don't think you can get ethics. I don't think you can get ethics on 7 days bed rest. Like that would be hard to get ethics approval for. You reckon?
But if you're fasted and bed rest,
yeah,
these days it would be hard. They've definitely done it probably in the past, but
that's an uncomfortable point that people don't talk about, is some of this stuff that we know and some of the research we have would not pass ethics today, right? And there's all sorts of stuff around war crimes and that sort of thing as well that informed a lot of what we know today. So it's uncomfortable truths.
I'm surprised that they even got this many people. So 13 people. It was, and they mentioned this was unpaid. So these participants were self-motivated, and they used the CGMs basically to look at compliance because their glucose levels should stay low throughout the 7 days because they were free living. They weren't in a metabolic ward being watched, supervised by these researchers. So it's kind of crazy that people were willing to do this.
It doesn't surprise me. I don't know when this paper was released. I need to double check that, but it doesn't surprise me in sort of 2025 that you get that. I reckon there's enough people. You just have to source them. It would be hard to source them in one spot, right? It would be easy to source them on the internet. I can find 13 tomorrow on the internet. It's just like really hard if they need to be in my city.
Right. Yeah, it was a 2025 paper.
Yeah. So there's some interesting stuff here that we didn't quite understand and some stuff that we did understand, right? So heart rate peak was slightly higher before the fasting period. That's probably to do with just like you can't sustain the intensity. O2 pulse, I can't work out exactly what this was, but this decreased 12%. I'm not sure how relevant that is, but the peak lactate stuff was interesting. So peak lactate and RPE was similar levels before and after fasting, but the respiratory exchange ratio went down. Yeah, so they had less carb burn in this context, right? So a higher respiratory exchange ratio was just more carbs burnt.
I don't think we explained that the study protocol being that they did this graded exercise test before in the fasted state. I think it was just like baseline fasted. And then after on day 5 or 6, or was it on day 7? Day 7 of fasting. They did that exact same graded exercise test to look at fat oxidation rates, carbohydrate oxidation rates, and blood lactate levels and VO2 max.
Yeah.
So the peak blood lactate concentrations were the same. RPE being the same seems interesting too, because you think it would feel harder after 7 days of fasting.
But the RPE is what stops the test. So it's the independent variable.
Yeah, I guess if it was the same absolute intensity, then the RPE would be higher.
Yes.
At the end.
So because these are subjects and they're volunteers, you can't force them to go at certain things. So the RPE is the dependent variables in this case. So that's the only reason it doesn't change. But otherwise, you'd be 100% correct. If we looked at RPE normalized to work rate, it would be up for sure. So they were burning less carbs here, the respiratory exchange ratio we talked about. But the other things, the respiratory exchange ratio also declined at rest, which would make sense. They're burning more fat, less carbs. Their blood pressure lowered, also very normal in this context. Their resting metabolic rate was unchanged. Doesn't surprise me. It's not going to change markedly over a week, I don't think.
I thought that was interesting. I don't know. 7 days seems like a lot. It seems like a lot for like your metabolic rate didn't change. It did not adapt to the lower. I guess maybe their energy expenditure was still enough that they didn't have to adapt to a lower energy expenditure.
Yeah, my gut is that they probably did less non-exercise activity, right? They stopped fidgeting, they stopped moving around as much. But I think probably post 7 days is when you'd start seeing maybe resting metabolic rate changes. But even then, I'm not too sure on this. I'm no fasting expert.
But it also isn't funny because like people always say like, oh, you can't skip a meal or like don't fast through the morning because you're gonna destroy your metabolic rate. This was 7 days and it didn't change. So yeah, I think we're fine.
We're not as fragile as people say. You certainly won't burn as much. I mean, there is a thermic effect of food. When you eat food, it takes energy to digest that, sure, but I'm not convinced that we're not that fragile. Mm-hmm. So resting lactate was higher. We were talking about this before. Resting lactate was higher, and you said this is something that your lab mate said is pretty normal.
Well, during exercise. So this difference, so they observed a significant difference in lactate. So their stats show this significant.
So statistically significant, maybe not clinically significant or meaningful, right?
So the resting lactate should be around 1 millimolar in a healthy individual. And they had 1.1 being the baseline, and then before fasting, and then after the 7 days of fasting, the resting lactate was 1.5. So that's a 0.4 difference in resting lactate. And I would put that within the error measurement of error. And I don't think that that's necessarily that relevant.
Yeah. That's exactly what I highlighted to you. I was like, is this real or meaningful? I've got no idea. I don't think it is, but interesting. And then just let's talk about that. You would anticipate seeing this in exercise, though.
So they observed higher lactate levels during the graded exercise test in the fasted state than baseline, which I can't really wrap my head around. And we were discussing this off air, that if you're increasing carbohydrate oxidation, or if you're decreasing carbohydrate oxidation, you would expect lower lactate levels. So they had higher fat oxidation rates during this graded exercise test, but they were producing more lactate, which either means that we're producing more or we're clearing less. And I can't really wrap my head around what's happening there, because you would even expect lactate clearance to go up in the absence of low carbohydrate availability, because even external organs like our heart and our brain, if glucose availability is low, we're looking for alternative sources. So I would hypothesize that clearance would go up in the fasted state.
So in that context, for listeners, we would take lactate and things like muscle breakdown. That's part of why we see muscle breakdown. And that would go to, and we would do what is called gluconeogenesis. We would make new glucose from those substrates so that we had some carbohydrates to use for brain, et cetera, et cetera, et cetera. So, it's a good question. I don't have an answer. I don't think Kristi has an answer. If you know, please let us know, because we're interested.
Because lactate goes up even in the rested state. If you gave a carbohydrate bolus, you would see a bump in lactate just postprandial, suggesting that you're increasing carbohydrate metabolism. So this is the opposite. This is in the fasted state, and then exercising, increasing lactate further. So
Watch
yeah,
this.
it is
Watch this
interesting.
space. When continuous lactate monitors get here, people are going to freak out that eating's bad for them because their lactate goes up. Um, I'm waiting for that. I'm waiting for that moment. But let's move on. I know you're a big mitochondria fan. You're studying mitochondria. There is a paper here, and I wanted to share some stuff. I really just want to broaden the listeners' understanding of mitochondria beyond the high school biology. Right, the high school biology is mitochondria, the powerhouses of the cell. So it's a good review paper here. So it's called Exercises Mitochondrial Medicine. How does the exercise prescription affect mitochondrial adaptations to training? I actually think I sent this to you when I found it the first time and said, hey, check this out.
Yeah.
It's a cool article. My favorite part of this article is the fun fact that they said the quote, energy producing powerhouses of the cell was an analogy first coined in 1957, and it has been propagated via copy and paste ever since, which is wild to me. But I think it's important here, like, beyond just powering the cell, what do mitochondria do?
Yeah, I mean, they're the primary site of energy metabolism, so that's no small feat of
Yep.
keeping us alive. But they also are involved in cell signaling, calcium handling, homeostasis, hormone synthesis, programmed cell death. So the cell signaling, I bet, is part of that
Yep.
programmed cell death or
Yep.
apoptosis. And then they serve, I guess, other functions that I don't really know off the top of my head, but they are this multifaceted organelle that aren't only responsible for energy metabolism, although all of those roles are linked to energy metabolism. So
Yeah. They sort of are involved. Like, so you handle calcium via energy is your point, and cells die because of lack of energy, et cetera, et cetera. So it's kind of, they have roles in all those, but it still fundamentally revolves around energy provision.
right, and like the byproducts of metabolism, energy metabolism, are the cell signals. So that's why they're involved in cell signaling.
Yeah.
So it all comes back to their energy metabolism.
Yeah, and so then there's also this view that these things look like kidneys with like a little kidney bean with like a little couple of like squiggles drawn inside them. That's how they show up in textbooks and that. But let's talk about it. They can look different. They can be different. They can fuse together. They can break apart. Let's talk a bit about that.
Yeah. But just to start though, mitochondria are very plastic. So they have a high level of plasticity, meaning that they change, they undergo dramatic changes in structure and function depending on the environment that we put it in. So our lifestyle, even down to diet, but mostly exercise. So this paper discusses exercise as mitochondrial medicine because exercise is the most potent physiological stimulus for changing mitochondria, and vice versa. Sedentary behaviors and inactivity also dramatically changes the structure, function, content of our mitochondria within our skeletal muscle. And mitochondria within skeletal muscle specifically is very important just because muscle is our metabolic organ that is largely responsible for whole body metabolism. So if we just keep breaking down the layer and get down to what the mitochondria, which mitochondria in what organ are really tightly regulating whole body metabolism, it is within our skeletal muscle. And that's what responds to exercise. So I think when we think of mitochondria, we say that term very broadly, but if we think about health and metabolism, it's skeletal muscle mitochondria to a large degree. So sorry, that was a ramble.
No,
I just wanted to throw that in there.
It's super important. I think we talked about on this podcast, and I certainly wrote an article on it, the mitochondrial transplants. So moving them around our own body or getting them from another organism and that sort of thing. And I sort of alluded to the fact that I think this is probably happening in some athletes. It's probably like, because it's untestable. It's their own mitochondria or someone else's. You can't really test for it. And it's certainly being used in mitochondrial dysfunctions, right, and disorders. So it's quite a interesting field here, but they can be harvested from fat or other muscles and those sort of things, right? So
that's crazy.
Yeah, it's really.
But yeah, so this paper basically categorizes, uses 5 different categories of how to frame mitochondrial adaptations, because it's not just what gets talked about a lot, especially within exercise physiology, is just mitochondrial biogenesis. So the creation of new mitochondria or the expansion of the mitochondrial reticulum, which, like you said, it's not just, we don't just have these small kidney bean like structures, we can actually just elongate them, and that is considered mitochondrial biogenesis. It's just making one mitochondria bigger. But this paper talks about 5 different categories to frame these adaptations. So they talk about increasing mitochondrial content. They talk about like the behaviors of mitochondria, so how they fuse together to elongate, how they break apart to basically cut one mitochondria into 2, and how they undergo mitophagy. So that's mitochondrial specific autophagy, so breaking down old, maybe dysfunctional mitochondria to create better new mitochondria using the building blocks. The way they function. So function is usually we talk about mitochondrial function in the terms of respiratory capacity. So how well can we produce ATP? How well are these powerhouses working for a given amount of oxygen? And then the activities of the mitochondria. So within a mitochondria, there's tons of different proteins, and most notably the electron transport chain. So that's where we actually deliver electrons, get passed along eventually to make ATP and power the cells. And so the activities of those enzymes can be measured, and that can change with exercise. And these are the plastic, like these are the features that are plastic, I guess. And then within that is also lastly the structure. So how is this actual morphology of the mitochondria changing and adapting?
Like classically, if you think about the kidney bean you see in your textbook, right? The kidney bean with a couple of things inside, we can increase the number of sort of squiggles inside of it. We can change the kidney bean shape. It can be longer, smaller. It can break apart if it's big, and now it's breaking into 2. An old broken one can die off, and those building blocks get used again, as you said. And all of this depends on the signals you're sending it. And those signals, the strongest one and the predominant one is exercise, high versus low intensity, strength versus other, etc., etc., etc. And then you can have energetic stresses, right? So do you have enough energy or not? Do you have enough carbohydrates or not? And then other aspects as well, part of your diet, right? Part of your diet, lifestyle, et cetera, et cetera, et cetera. So we are sending all these signals at all times to all cells in our bodies. Mitochondria are no different, even though mitochondria historically were actually something we ingested, right? So they are not of human origin. I don't know if you know the story, but they are a secondary organism that we ingested and became symbiotic with us. And our mitochondria that we have is directly from our mother. So we have our mother's mitochondria.
You say ingested as if we ate a mitochondria. I think it's just eating. Didn't like a cell engulf
the
mitochondria?
Correct.
There's no chewing and swallowing of that mitochondria.
Well, some people just engulf. I don't know. We're probably eating mitochondria as well. Who knows? But it's it's a good point. It was way before we probably ate as cells, and more before when it was sort of single cell organisms. But it's an interesting thing is that these things are older than humans are.
Yeah, and another fun fact: mitochondria have their own genome.
Yep.
So they encode 13 genes.
Yeah, I'm not sure, but all of that mitochondrial DNA is from your mum, though. You get 0 from your dad, which is cool. So exercise variables and changes to mitochondria. So let's talk about this. So, like, what are the variables and how do they impact mitochondria? Like, are we going longer? Are we going harder? What happens there with our mitochondria? Because there's classic statements that float around is like volume does this and intensity does that, and I think it's a bit reductionist from what I've read.
Right, and so again, this paper. They looked at exercise prescription using the FIT VP framework, which is frequency, intensity,
time and type.
What is it? Oh yeah,
time and frequency, type, intensity, time, type,
and then volume and progression. And so, how do we use those to understand all of these 5 categories of mitochondrial adaptations? And I mean, we'll start with the first, just being that mitochondrial content seems to be driven by volume. So takeaway from this paper is that volume drives increases in mitochondrial content, and that plateaus, I guess, at a certain point. We don't just keep increasing mitochondrial content. Is that?
Yeah, it seemed to look like there was a bit of a threshold phenomenon or maybe a ceiling phenomenon whereby if you went high enough in volume, you got sort of most of this and then it very much plateaued from there. So it's not exponential or it's not even linear. It's like up to a point, volume is important, and then it sort of seems to plateau out.
Yeah. And then an important point that I really love that they pointed out in this paper is that there might be a intensity threshold for which volume accumulated above increases mitochondrial content, but where increasing duration below this threshold can't compensate for a low exercise intensity. So even if you're doing, they threw in an example of a study where 6 hours a day for 32 days of very low intensity exercise, there's no increase in mitochondrial content, suggesting that you have to accumulate volume above a certain threshold, which I personally would think would be above our first metabolic threshold or our first lactate threshold.
Yeah, I tend to. It doesn't surprise me if you think about it. Otherwise, we'd be accumulating mitochondria walking about, right? Like
Yeah.
it's not. It needs to be stressful enough for us to actually like adapting to exercise is a energy costly event. Creating new things is energy costly, and you don't want to be doing that if you don't need it. So if you can already compensate, if it's not challenging, you're not going to need to compensate, right?
Right.
So it needs to challenge you to change you, right? To use one of those fitness isms.
Yeah. So volume above a certain threshold, accumulate. And then I guess if you can accumulate as much volume near high intensity,
Yeah,
then that seems to be driving greater adaptations.
and remember again for athletes, right? People who are not just trying to win the award for the most mitochondria or the best working mitochondria. There are other considerations here. It's not a mitochondria problem. We don't measure mitochondria on the start line and give out medals. So this is part of the equation and it's part of our understanding, not a prescription for training. Awesome. Anything else you wanted to? Any other takeaways here in terms of like things to think about for mitochondrial adaptation?
I don't know. I just thought that some of the things were interesting. That like the Kristi, so the squiggles within the
Yep.
mitochondria, that's where our electron transport chains are located. So the more
Yep.
of that, hopefully, the more enzymes we have, the more respiratory capacity we have, and those things can, I guess, change without increasing actual mitochondrial content. So if we're just looking at increases in mitochondrial content, that's not taking into consideration any changes in function. And so they talk about that, the authors of this paper talk about how even just like sprint interval training can increase respiratory function without necessarily changing content, which I think is interesting. And that has an actual real life takeaway, being that if your function is increasing, then maybe your health and performance is increasing without more mitochondria. And then I also thought it was interesting that they talked about that density could be higher in athletes and challenging what they thought before, that you can't change, I guess, the surface area of our mitochondria or the density of it, but that athletes actually do have higher crystal
Yeah.
density.
Yeah. It's an interesting thing, like because I guess the question is how plastic is that? Like, have we seen changes in that? And this is part of the challenge we have is like are you a good athlete because you have better mitochondria or vice versa? Do we train that? And we think it's trained, and I think there's enough evidence to suggest that, but it is always hard, particularly with mitochondria, because a lot of the ways we test them or measure them are very imprecise. We look at mass or we look at the amount of an enzyme, and you're like, that doesn't really tell us about individual changes in individual muscles or individual mitochondria. We're just getting a global picture. So it can be a little bit difficult for us to understand some of these changes and even measure them, right? And I think if anything, hopefully technology allows us to do that. I mean, you also work in glycogen, or you've done a bit of work with glycogen in that sort of muscle biopsy space. Like a muscle biopsy measures glycogen content, right? All right, we got glycogen content. Problem is we can't sample that same space again because we've taken it out. So we are hopefully nearing a space where we've got better measurements using advanced imaging techniques to understand mitochondria, glycogen, all these things to really to change the way we think about them. Because I think we have a useful enough model for how these things work at the moment. I don't think we understand them properly.
Oh yeah, there's still so much to learn.
Yeah.
Another point, though, they also looked at resistance training studies. So they looked at any type of exercise intervention that measured any sort of mitochondrial adaptation in this paper, and uh, like basically, I guess the takeaway is that obviously endurance training is going to lead to greater mitochondrial adaptations than resistance training, and the results from resistance training may be that mitochondria don't necessarily change in content or function, and it's actually just that hypertrophy is increasing the amount of muscle there, so it's kind of diluting the mitochondria within the muscle, but that it's not that mitochondria is not, it's not that mitochondria content is decreasing, it's just being diluted by the increase in muscle mass. And there might be a slight increase in mitochondrial content with resistance training, but that endurance training is of course going to come out on top because it's more energy demanding, and they're the powerhouse of the cell.
Yeah. Well, this is the other challenge is like we tend to, again, thinking about exercise as like anything, it's like there are thresholds. So there's like a spectrum of exercise, right? So there's true strength training, 1 rep max squat, 1 rep max deadlift, which is not the same as reps of 10 on something else, which is not the same as like a CrossFit workout, which is more hybrid again. And then there's more running based. So like it's really a spectrum to think about across that. Like 30-meter sled push, 60-meter sled push, 100-meter sled push. Those are increasingly endurance-based activities. So it's worthwhile considering it more as a spectrum like that, rather than just like binary. It's like, oh, it's got a weight, so it must be strength, so therefore it's terrible for mitochondria. It's like I always say this is physiology is dimmer switches. It's not on-off switches. It's like more and less. It's not all or nothing. So it's always little bits and little bits. But Kristi, this has been great. We've appreciated having you. Thank you for filling in while Xylon was away in Rwanda. So we do appreciate you, and thank you for co-hosting. You did really well. And we are going to keep in touch with you. You're going to come back on when you finish more studies in your PhD. You're going to maybe co-host again. We might ask for another video of your piano playing skills.
I need a new challenge. What's the application requirements for the next one?
Do some training for High Rocks. How's that? Or is that non-palatable? But no, we are interested. Keep us updated how you go in Toronto. We will be following along. We'll follow along with that. But thank you so much, Kristi. And thanks to all the listeners who tuned in this week. Please do like the podcast, share it with somebody if you can. Of course, subscribe to the podcast, follow us on social media, join our Strava club, whatever you'd like to do. We'd appreciate you there. We're on YouTube as well. So thank you, Kristi. You have a great day.
No, thank you so much for having me. And everyone, vote if you want to see me back on here again.
Okay, well, yeah, let us know. Send us a DM, send us an email if you want to see Kristi back on, and we'll go from there.